Hello everyone. So what I'm gonna do in this recording, I'm gonna show you how you can do the analysis for P53, something we went through in the lecture, but I realized it was not recorded. So I'm gonna show you what you have to do with the sequences that are given to you from the website, P53 protein sequences. And then you can do that by yourself, adding the sequence that you were assigned, and the same thing you'll do for the mitochondrial DNA. So what do you have to do is two things using the MEGA11 software, which again is a compiled software that compiles a series of different access to different algorithms here. Aligning algorithms and so on, phylogenetic algorithms, something that allows you to figure out the relationship in terms of time between different organisms and so on. What we're interested on is the align algorithm here and the phylogenetic algorithms, I should say, that are available from this tool here. So what we're going to do first, we're going to do the alignment. And in order to do that, you have to click here on the alignment. You're building an alignment, brand new in this case. You're not using a previous one. And what it's going to ask you in the next window here is whether you want to create a new alignment, open something that you saved before. We are creating a new one, so we're going to click on the top right there. Next thing is going to ask you is what type of sequences are you working with. So when you work with mitochondrial DNA, you use DNA. In this case, we're working with protein sequences, P53 protein sequences, so we'll click on proteins right here. And then it's a matter of adding your sequences to this window and run the alignment algorithm. So the sequences are those that were posted here, plus the one again. You will need the one assigned to you. I'll repeat it over and over. So I just copy them from that file right here. Just click copy. And then I'll paste them here. Your sequences have been entered, but they have not been aligned yet. So what you have to do next is to align them with the mouse. So you can either select all the sequence directly, or if you don't do that, it's going to remind you to do that, the software. So now there are different algorithms that can be used. Common one, Clustal Omega, and the other one is Muscle. Muscle is the most commonly used nowadays. So we're going to use the muscle one there. You click and it's going to give you a window with all the possible options you can change. Gaps, the extension, gap extension, and so on. This is to create basically the relationship between the different organisms in terms of time, which one you want to use as an algorithm. Just don't change anything, just execute orders. Otherwise, you're going to mess things up probably. Or if you want to play around, you can even play around with this stuff. But this is not the point of this exercise, lab exercise. So press OK here, and then it's going to align the sequences for you. Nice and easy. In this case, it's going to do it fairly quickly because you're dealing with only a few hundreds letters ultimately. Whereas when it comes to the mitochondrial DNA, it might take several minutes to do that. So be patient and wait around, do something else in the meantime. So here are the sequences that have been aligned. Having this color pattern for the same amino acid types and so on really helps to see the alignments instead of just looking at blunt letters there. And you can start seeing how ultimately the phylogenetic algorithm works is ultimately by comparing these sequences. These are the best alignments, so this is the one with the highest scores, of course. It's going to look basically for common sequences or differences where there is insertion, deletion, and so on. Here you can see a deletion in Macaca nemestrina here, a very long deletion, actually it's multiple deletions. If you look, there is one deletion there is another long deletion in this part right here, and so on. And then there are some substitution that I was pointing to you during the lecture, like this substitution right here, this 3 amino acid in this group of primates compared to the other ones. And clearly these are events that most likely have generated this divergent evolution of these organisms. They're commonly found. You can see there are multiple ones unique to three different species, Papia nubis, macaca, and so on. These two monkeys of these two primates of the same genus. This one right here includes also lemurs, which have split before. If you look at an old evolutionary tree, you'll see that lemurs actually have split from the rest of the primates earlier. The macaca and Papia nubis. So you can see this was probably an event common to all of 4 different species, whereas the next one that I was pointing to you was unique only to Papia nubis and the 2 macaca species there. So that's what basically the phylogenetic tree algorithm is going to use later on to create the graphic representation that we know as phylogenetic tree. So once you have all these sequences aligned, now all you have to do is actually save it. We're going to export it in the MEGA format. And then we're going to retrieve them later on. So I'm saving them on my desktop. I have already saved them before, but I have a file that says, well, it's primates, P53 sequences in the FASTA format that have been aligned. So I'm going to save it. Doesn't matter what you write here, but doesn't really change much. Saved these sequences, you can actually close this sub window. This is just a sub window of MEGA11, specific for the alignments. You will close it. And now we're going to open a second sub window, which is the one that connects you to the algorithms that do generate the phylogenetic tree by clicking on this tab right here, phylogeny. And now you can choose the algorithm you want to use. You can even test them all if you want to play by yourself. Here we're just going to use this one right here, the neighbor joining tree algorithm. And all we really have to do now is to upload the sequence that we saved before in the MEGA format, which is the one right here. Now it's going to ask you if you want to change any of these parameters here. Don't do it as we said before. You can play around if you want. Many things that are in there. Again, this goes beyond the scope of this lab. All we want to see is really how you can use these sequences to generate these phylogenetic trees. And so press OK here, and it's going to give you ultimately the phylogenetic trees. It's opening the sub window, which shows you the relationship between these different organisms. Now here you can play with different things. The way you can order them, you can either show them by input order and so on. Just are very little things. The layout is something that I like to play around with. So if you say auto size, is actually going to show you a little bit larger the windows in a way that also makes it more visible the relationship between different organisms. For example, what I can see here, the palm troglodytes and pampaniscus, so chimps and bonobos, are very closely related through this common ancestor, which have split from Homo sapiens, from yourself and myself, at a certain point in history. Again, what I was pointing to you in the lecture is how using proteins ultimately has a big limitation in terms of defining events that have happened fairly recently in evolutionary times. Here we're talking about a few million years when Homo sapiens and pampaniscus and pantroglodytes have split it. And you can see how with the protein analysis, there is a very hard time to define time. This is not really time, but it gives you a sense of distance, which is proportionate to time. Something that you will see with the mitochondrial DNA analysis instead, which is much more clear and much more defined instead of what you get with the protein. But you can get a sense of the relationship here, how, for example, Homo sapiens is more closely related with chimps and bonobos than with the gorillas. And even further, how much more earlier in time it actually split from oh my gosh, sorry, but my brain is fried. I'm recording this right after the lecture. So oh my gosh, okay, I'll tell you later on. It's cute orange organism here. My brain is completely fried here. Okay, so and as I was mentioning before, the lemur actually in the primate order, at least those that you are analyzing, is the one that split the earliest compared to all the other ones right here. And I was pointing to you how baboons and macacos here are actually much more closely related. That's where I was showing those three different amino acids compared to the other one. Okay. So orangutans, sorry, that's what I was talking about here. These are different species of orangutans, parts of the same genus Pongo. Okay, sorry, my brain is completely fried by now. So I'm actually gonna stop right here. So other thing, sorry, that you can play with here is you can play with the graphic part. What you have to do, ultimately keep in mind, you have actually to screenshot these and add it to your assignment for both the P53 and the mitochondrial DNA, again, with the additional sequence that was assigned specifically to your group. You can you can even generate the trees in different ways if you want, like a circular way. So here you can make them circle, not very helpful to look at what we're saying here, straight lines, as you might be used to see sometimes online. I like it personally, even better than the rectangular one, but you can play around and show it in different ways. Okay, so there are different options. You can practice with this part on your own time. It's not the scope here to show you, okay, just click this and click that, but you can see all of this stuff, how it's can allow you to do this stuff. Here, if you want, you can actually separate them more or less, the different species, if it helps you with the screenshot. That's the only thing I show you. All right, guys, I'm stopping right here. I hope this helps you with the generation of your trees. Again, there's gonna be some additional sequence I'm gonna add for another two different, or at least one more tree that you'll have to generate in your assignment. All right, bye everyone.